The exact tools and software that you're going to need for being a biochemist or a molecular biologist is going to depend on the type of research that you're doing, but these are some core programs that everybody should familiarize themselves with because you're probably going to be using them. So just a quick overview, and then will post a link to a version with links so that you can then go and download these things and check them out. So let's start with databases. So the core ones, if you're working with proteins, UniProt, that's your first go-to place. It's got the information about basically like every protein. It has a lot of links to take you to other sites, but here you can find proteins from all different species. You can compare these proteins, learn about their sequences, get links to their structures and that sort of thing. And so those links to the structures are going to take you to the PDB, so the protein data bank that has all the structures that have been solved from things like X-ray crystallography and cryo-EM. If you want to then view those proteins, you can use a structure viewer like Chimera X or Pymol. These are really great for allowing you to interactively work with these 3D protein models and then make really pretty figures. If you want to figure out more about like the makeup of the protein, there's this tool called Expasy ProtParam that's really helpful for finding things like the extinction coefficient, which is going to allow you to figure out the concentration of a pure protein based on its absorbance of UV light. It'll also tell you things like the PI, the isoelectric point, which is really helpful if you're trying to purify the protein and want to know if it's like positively or negatively charged, because you're trying to purify it with ion exchange chromatography, which the kind that you use is going to depend on whether your protein is positively or negatively charged. And so that's where you go to the PI and then the pH and that sort of thing. Okay, so for nucleic acids, your go-to sources, GenBank, this is like all the sequencing data, really awesome. Then it also has links to other tools, so this is through like the NCBI, and they have things like RefSeq and stuff, which has the reference genome, and there's various technical things and more in other posts. But GenBank is going to have like all the public sequencing data. BLAST, this is really great if you're trying to search for a nucleic acid to see you have some sequence and you want to see where it comes from, or maybe you're trying to design primers and you want to make sure that those, for your PCR, you want to make sure those primers aren't going to bind places that you don't want. GenBank is really great. BLAST is really great for that. You can also do things like compare sequences to get like evolutionary information, as well as they have things where you can go from compare protein sequences as well as protein to DNA and all these various tools for things like that. Speaking of cloning and like PCR and stuff, if you're wanting to work with plasmids, so like circular pieces of DNA, you're going to need to design your cloning strategies. You might be using like PCR, you might be using restriction enzymes, but you're going to need to know the sequence of those plasmids and be able to work with them. And so you're going to want some sort of plasmid viewer. So a common one that's used is SnapGene. There's SnapGene Viewer, which is a free version, and then there's like a paid version. So you can't do as much with just the viewer, but if you just need to view a plasmid, it has a nice interactive interface. I used CLC Main Workbench in my last lab. That's one that you have to pay for, but it has some nice features. A totally free one is Ape, a plasmid editor, and they have some nice videos and stuff on YouTube as well. There's also DNA Star, which has some free version, and Benchling, which also Benchling is helpful for like e-notebooks. So speaking of e-notebooks, that's, you don't need an e-notebook. Well, your lab might make you, but so some labs, everybody uses the same one. So this might be Benchling, which is like cloud features and things so that people can share notebooks and share protocols and that sort of thing. You might just have your own personal notebook. So I use EndNote to keep a virtual notebook, and I also keep handwritten notes so that I type up my handwritten notes into my virtual notebook, and so I can cross-reference things and I can search my notebook, so I find that really helpful. Okay, so a couple of other things. So in terms of staying organized, you're going to also want to make sure that you organize your references. So when you find papers, you want to be able to then know be able to locate those papers again, and then when you're writing a paper or that sort of thing, be able to cite them. So be able to give those authors credit. So you're going to want to use a reference manager, something like Mendeley's, Zotero, EndNotes. There's something now called like ReadCube, I think, Pages. There's various reference managers that you can use in order to keep track of your references. And so every time you read a paper, I recommend saving that reference into your reference manager. Even if you don't think you're going to ever need that paper to cite it or anything, you want to make sure that when you think back later, oh, I know I read that somewhere, you can go and find that source for the paper. Okay, you also want to be able to find papers in the first place. So the papers are coming out all the time, and you need to be able to stay up to date. The main places where you're going to find scientific papers, so there's this thing called PubMed, which is this database which has like all the information about all of the published papers and links to the publisher's website where you can download those papers. There's also like PubMed Central, which has all of the like openly accessible papers so that are free to read, nothing behind a paywall. So there's also bioRxiv, which this doesn't have published papers, this has preprints, so things that haven't gone through that peer review process yet, but have been posted on bioRxiv, and this is another common place where you're going to find papers, and these are going to be really, really hot off the press stuff. In order to stay up to date with all these papers and preprints that are coming out, you might want to look into setting up RSS feeds and using an RSS viewer. So bioRxiv, all these different publishers, even PubMed, you can set up alerts and you can set up RSS feeds, which are basically like these custom feeds when there's new articles come out on a specific topic that you specify, then it'll go to this RSS feed, and you can use an RSS viewer where you can then put these feeds together. And so these are things like I use NewsBlur. There's also Feedly, the Old Reader, InnoReader, etc. And this is going to help you stay up to date. And you can also set like alerts on PubMed, alerts on Google Scholar, this sort of thing. This is really, really helpful for making sure that you're not missing papers as they come out. So you can set like key search terms or keywords or key authors, and then if those people publish something, if those terms come up, then you'll get an alert. Super duper helpful. Okay, just a couple last things. You're going to probably want some sort of vector graphics program and to get used to using that vector graphics program. So this could be like Adobe Illustrator. A free version is Inkscape. These are things that are going to allow you to make graphics and figures, not just for fun stuff, but also for like serious stuff, like making figures for papers or for presentations. The really nice thing about a vector graphics program is it's working in like points and things. It's basically telling the computer how to draw the figure rather than being an image. And so you're not going to get it. You can save it in ways and blow things up that they're not going to get all blurry. And so if you're giving a presentation, you want to have your figures in this high quality form, so maybe something like a PDF, where you can blow it up and it's not going to get all pixely, as long as there's not actual pictures in there, which would still get pixely. But the words and the lines and all of these things will stay nice and sharp. Okay, also another helpful thing is to learn a bit of coding, and so you're most commonly probably you're going to be wanting to learn Python. So I have a couple posts on why it's important to learn Python, as well as some basic tips for using Python. I like to use Jupyter Lab and Jupyter Notebook, which are basically these interactive coding tools that you can use on your computer, and you can share files with other people. These are interactive notebooks, so you can be looking at the code while you're making it. You can then like run little snippets of code at a time, and it also has a nice interface that allows you to easily see different features and that sort of thing. So much more on it in other posts, but I recommend that. So there's Jupyter Lab and there's Jupyter Notebook. So Jupyter Notebook are like the Python files that have these interactive Python files, but then Jupyter Lab can also work with other types of programming languages, as well as like just core bash and stuff, so terminal type stuff. Basically it can do more than just the Jupyter Notebooks, but it includes the Jupyter Notebooks. And also you might want to learn a little R. If you're working with R, I recommend RStudio, which is a desktop application that you can use in order to interactively work with R, which is another programming language. It's used commonly in things like ecology, various things like that. And so much more on those in other posts. But those are the key tools that you will probably want if you are a biochemist or training to be a biochemist or that sort of thing. And then you're going to need to pick up more tools based on what your project is. And I will also link to a post I have with more databases and tools. For some of them are like much more specialized. But I hope this helps give you an overview of the type of things that you should be getting familiar with if you are starting out on the journey of becoming a biochemist. And so I wish you all the best on the journey, because it's really, really exciting. And so hopefully this isn't overwhelming. It's meant to be helpful. And so hope it helps.